Fermentation, Part F of the Carbohydrate Catabolism Lab. Introduction to this experiment. Anaerobic respiration requires the electron transport chain. However, it does not require oxygen. In aerobic respiration, oxygen is required. But in anaerobic respiration, molecules such as nitrate are used as the final electron acceptor instead of oxygen. Fermentation takes place in human cells, and it also occurs without oxygen, and this is a way human cells can produce ATP without oxygen. Fermentation occurs in the absence of oxygen when organisms can only use oxygen as the final electron acceptor. So in human cells are not able to use nitrate as the final electron acceptor. So human cells rely on fermentation. And this makes ATP via glycolysis. And the 4 ATP are made, but 2 of them are used, and this is substrate level phosphorylation. Muscle cells produce lactate or lactic acid, and yeast cells produce ethanol or alcohol. Glucose, in this diagram you can see that they're showing glucose as the only sugar here, but other sugars such as fructose can be used in fermentation also. And here you can see the 2 divisions of fermentation, lactic acid and alcohol. So alcohol fermentation is ethanol. Fermentation produces far less ATP than oxidative phosphorylation and the normal process of cellular respiration, which produces 30 to 34 ATP. There's a significant amount of NAD+ that needs to be regenerated to run glycolysis. This is because there's an excess of NADH, and this excess of NADH needs to be reduced to NAD+, and the fermentation allows for this to happen. Otherwise, there would be a imbalance in the NAD+ and NADH ratio. Humans use fermentation in baking bread, which causes bread to rise. The carbon dioxide released from fermentation causes bread to rise. Humans also use fermentation in wine making and beer making. Enzymes play major roles in cells, as we know. The reactions of cellular respiration and fermentation use enzymes. Kinases are involved in the transfer of a phosphate from one molecule to another. An example is a hexokinase. The hexokinase is seen in the first step of glycolysis as we add a phosphate to glucose to make we use hexokinase to make glucose 6-phosphate from glucose by adding the phosphate. Dehydrogenases oxidize a substrate. An example is G3P dehydrogenase. And this enzyme oxidizes glyceraldehyde 3-phosphate to make 1,3-bisphosphoglycerate. Enzymes can be regulated, as we know. Temperature affects enzymes, pH affects enzymes, inhibitors affect enzymes, salinity affects enzymes. And enzymes are one way cellular respiration and fermentation are regulated. An example is phosphofructokinase, which is one of the most important regulatory enzymes. This enzyme controls the irreversible step. In the whole cellular respiration aspect, there's one step. Once this reaction takes place, there's no going back. And this enzyme controls that. This matters because if there's enough ATP in the cell does not need to produce more ATP, so phosphofructokinase will stop allowing cellular respiration to continue at that rate. It'll slow the rate down. But if there is not enough ATP, phosphofructokinase will accommodate for that. Phosphofructokinase contains an allosteric site, which is different from the active site. And this is where ATP will bind to show that there is enough ATP in the cell. And this binding will regulate the process. Our hypothesis, tube 2, which contains only glucose, will produce no carbon dioxide because it does not contain yeast. Yeast fermentation will produce carbon dioxide as a waste product, and there is no yeast, so this carbon dioxide will not be produced, and this is the negative control of our experiment. Tubes 1, 3, and 4 each will ferment, but at different levels, and we'll measure this by measuring the amount of carbon dioxide produced. The procedure was to add yeast to beakers 1, 3, and 4 and stir. Beaker 2 again, we did not add yeast. Add the mixtures from each tube to its respective swan tube. The swan tubes allowed for us to measure the CO2 production in its unique shape, and we incubated each swan tube at 37 degrees Celsius for 1 hour. We recorded the size of carbon dioxide, the carbon dioxide air bubble after 30 and 60 minutes. These are the swan tubes, labeled 1, 2, 3, and 4. And as you can see, there's no air bubbles to begin. These are the swan tubes after 30 minutes of incubation. As you can see, there's a pretty decent sized air bubble on tube 1, no air bubble on tube 2, another air bubble on tube 3, and it looks like a tiny air bubble, but I'll explain why that is false, a false positive later. And again, air bubble on tube 1, nothing on tube 2, a big air bubble on tube 3, and what looks like an air bubble on tube 4. However, if you examine closely, this air bubble is just happening, and this air bubble, in this picture it looks like there's an air bubble, but when you examine it closely you can see there's liquid on the top, and because of natural factors the solution is pushing down, sort of causing this, and it's not an indication of fermentation. So as you can see, our glucose and yeast at 0 minutes, or actually everything at 0 minutes, had a carbon dioxide air bubble of measuring 0 millimeters. After 30 minutes, the glucose and yeast had an air bubble of 22 millimeters. Glucose itself, as we predicted, as our negative control identified, had no carbon dioxide air bubble. Fructose and yeast had an air bubble of 18 millimeters. Lactose and yeast had no air bubble. After 60 minutes, 28 millimeter air bubble on glucose and yeast. Again, as our negative control predicted, 0 millimeter air bubble for just glucose, no yeast. The fructose and yeast had 32 millimeter air bubble, and lactose and yeast had a 0 millimeter air bubble. This graph shows how tubes 1 and 3 were efficient in fermentation, in this case yeast fermentation, and they produce carbon dioxide. And over here you can see how much carbon dioxide is produced by measuring the millimeters, and as time passes the carbon dioxide produced increases. So based on this information we can draw a few conclusions. Glucose and fructose can both be used in fermentation. Lactose was not a usable sugar for fermentation, and tube 2 was our negative control as it did not contain yeast. Thanks for watching.